Antibody-Drug Conjugates, ADCs, represent an innovative approach to targeted cancer therapy. In this system, an antibody specifically binds to cancer cells, delivering a cytotoxic drug directly to them. Here's how it works. The ADC consists of three essential components. Antibody: this is the protein that targets a unique antigen present on the surface of cancer cells. Cytotoxic drug: this is the component that kills the cancer cells once delivered. Linker: this chemical bond attaches the cytotoxic drug to the antibody. Promising experimental drugs. Among the experimental ADCs, 3 drugs stand out. Patritumab Deruxtecan, primarily used for gastric and non-small cell lung cancers, this drug has FDA approval. The drug offers around 9 months of progression-free survival for gastric cancer and 8 months for lung cancer. Given the aggressive nature of gastric cancer, this is significant. Importantly, the drug is mostly well tolerated with minimal adverse effects. However, it's effective only when the cancer cell expresses the HER2 antigen, which also makes the cancer more aggressive. Over time, cancer may develop resistance to this drug. This medication is typically reserved for aggressive and treatment-resistant forms of the disease due to concerns about long-term safety and toxicity. Its high cost also limits its availability for early-stage diseases. Ifentimab Deruxtecan, currently in phase 3 clinical trials, this drug is used for multiple myeloma and has shown a 61% response rate, meaning it's effective in 61% of patients treated. Reludotatig Deruxtecan, this drug is designed for small cell lung cancer, showing about a 50% response rate and 6.5 months of median progression-free survival. Considering that small cell lung cancer has a poor prognosis, these results are encouraging. All 3 drugs share the common cytotoxic component, deruxtecan, considered a potent chemotherapeutic agent. While generally well tolerated, some serious complications like myelosuppression, interstitial lung disease, and Stevens-Johnson syndrome can occasionally occur. Clinical trial results. Studies indicate strong outcomes for ADCs. For instance, one study showed that 20% of patients were completely responsive to the treatment, essentially being cured of cancer. Another study found that 28% of patients with multiple myeloma treated with Ifentimab Deruxtecan had complete responses. Other ADC drugs like Adcetris and Enhertu have complete cure rates of 66% and 55%, respectively, in certain conditions. Future prospects. Several technologies are being explored to overcome limitations, such as drug resistance. Gene editing. Technologies like CRISPR-Cas9 aim to correct genetic mutations that cause resistance, particularly in HER2 gene-related breast cancer. Nanotechnology. Researchers are developing nanoparticles that can more effectively deliver cancer drugs, thereby minimizing damage to healthy cells. Immunotherapy. These drugs use the body's immune system to fight cancer and are now being combined with ADCs to enhance effectiveness. In summary, ADCs have shown considerable promise in treating various forms of cancer. While there are limitations, including drug resistance and high costs, ongoing research offers hope for more effective and accessible treatments.